The previous lectures have focused on action potentials. Action potentials are important because they are used to help a neuron communicate with another neuron. This communication occurs at a synapse, and oftentimes it occurs at what is called a chemical synapse. So our topic and outcomes are related to a synapse. Remember, these outcomes are what exam questions are based off. If you don't understand an objective or an outcome, or however you want to describe these, please ask. You've got SIs, you've got grad students, and you've got me. So to start off, we needed to define synapse. A synapse is a junction between a neuron and another cell. And there are two types of synapses, electrical and chemical. An electrical synapse is where cells are in direct contact with each other. Thus, when one cell becomes depolarized, all of the other cells it is in contact with become depolarized as well. An example of this is in your heart. These electrical synapses allow all the cells in your heart to contract at the same time. A chemical synapse, on the other hand, is where there is an actual gap between two cells. Consequently, a neurotransmitter that is released by one cell has to travel across the junction and bind to a receptor on the second cell. This is the type of synapse we will be discussing throughout the rest of the video. So a chemical synapse is made up of a presynaptic and a postsynaptic neuron. An action potential travels down the presynaptic neuron. This will eventually cause vesicles, which are right here, which are just containers inside of a cell, to move to the surface of the neuron where they release their contents. In this case, vesicles contain a chemical called a neurotransmitter. The neurotransmitter, which is these little dots down here, diffuse across the synapse via flow-down gradients and potentially, hopefully, bind to a ligand-gated receptor on the postsynaptic neuron or postsynaptic cell. This will cause ion channels to open, which will either cause or prevent an action potential from occurring. So to provide a little more detail, an action potential travels down an axon, and as it does so, it depolarizes the cell membrane. This depolarization will cause voltage-gated calcium channels on the axon terminal, or the end of the axon, to open. Since there is more calcium outside of the cell than inside, calcium will flow down its gradient and enter the cell. Calcium is essentially the green light or signal for something called exocytosis to occur. Exocytosis is the movement of the vesicles, those containers, to the cell membrane. Once the vesicles reach the membrane, the neurotransmitter is released. The neurotransmitter then diffuses across the synapse and potentially, not always, because it can just diffuse away, and we're going to talk about that on the next cell, but hopefully it binds to a ligand-gated channel on the second cell. Okay, so there's several steps that are going on here. If any one of those steps is eliminated, then the signal from the neuron or from the first cell or the presynaptic neuron is not going to get to this postsynaptic cell. Now, after a neurotransmitter is released, it is important that the neurotransmitter is removed from the synapse and receptors. This prevents a sustained signal from being produced. And this removal can happen in several ways. First, the neurotransmitter can just diffuse away from the synapse. The neurotransmitter, instead of binding to a receptor, can just be taken up or diffuse into the bloodstream and go away. The neurotransmitter can be broken down by enzymes. One of those enzymes is something called acetylcholinesterase. Whenever you see the suffix ACE, A-S-E, that's usually an enzyme that breaks something down. The neurotransmitter can be resequestered or taken back up into the presynaptic neuron, or it can also be taken up by another cell. Okay, and this is important, like I said, because it prevents a constant signal from being sent. Now, there are times where that we want that to happen, and we're going to talk about how that does happen later. But no matter the signal, no matter the strength, eventually you're going to want that neurotransmitter to go away. And so these are 4 ways that can happen. So this marks the end of week 1 of the nervous system. Are you able to do the following after watching this video? Can you identify presynaptic and postsynaptic cells? Can you describe how one neuron communicates with another cell? And can you explain how and why neurotransmitter is removed from the synapse.